This is a big smile.
All right, before we get started, I need audio. I need visual. How's the sound? How's the... No, don't touch that. How is the audio feed? How's the video feed? Yasmin, stop. Hit me in the chat, somebody. Give me audio, give me video. Am I feeding well? How's the sound? Up, Dory says we're good, Baba. Whoop. Good and good. All right, we are set. All right, Yasmin, we're live. We're live streaming on YouTube. See you? That's you.
No.
Jack, what's good, man? All right, Stephen, glad the sound is good. Thank you for letting me know. Let's just wait for some people to pile in here. We've got, looks like 5 right now. That's a good start. We'll see if anybody else comes in here. Why are you covering your face? Say hi to all of my friends over there, Yasmin. Say hi. Say hi. Can you guys say hi to Yasmin? They're saying they're going to say hi, Yasmin. Yasmin. Hey. 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 See, Sabrina says hi, Yasmin. Say hi. Say hi, Sabrina. Say hi, Sabrina. Say hi, Dory. All right, Yasmin. You're going to have to leave now because you won't say hi. Aw, Caroline said hi, Queen. That's nice. They love you. Say thank you. Say thanks for saying hi. All right, Yasmin, you have to go since you're not saying hi.
No.
You have to go now. No yelling on live stream, all right? Stay right here. Yasmin, I can't. Stay right here. All right. All right, y'all. I don't have much time. Little something came up, so this has to be pretty quick. I can give you guys about 20 to 30 good minutes of questions. Any of the questions that you guys have, I'll do my best to answer them. Just go ahead and put them in the live chat. Please be as specific as possible. If I don't understand your question, I'll just ask you to repeat it with any details that you... Yasmin, stop it. Hey, you're doing that on camera? What are you doing? Get out of here.
No.
Yasmin. All right. So whatever questions you guys have, I'll try my best to answer them within the allotted time that I have. So the time is now 7:43. We'll see how the questions and answers are going after 8 o'clock. I'll answer them as long as I can. So we're ready, right, Yasmin?
Yeah.
Yeah, we're ready. Yeah. All right, bring them in. Sabrina says, why is there 2 pathways of fermentation? So it really just depends on the organism. So in the case of like yeast, they ferment anaerobically using alcoholic fermentation. And there's different types of bacteria that ferment using lactic acid fermentation. They're just different pathways utilized by different organisms because they produce different enzymes. That's essentially what it does. Really, the only key difference between the 2 fermentation pathways is, if they're producing products of lactic acid or alcohol and CO2. That's pretty much the only difference. So that's why there's 2 pathways. Yeah. What are you doing? What are you eating your tongue? That's gross. All right, good question, Sabrina. Anybody else? Let them fly. I can handle more than one question every couple of minutes. Go ahead and send them, guys. I'll keep pace inside the live chat. You see all their questions here, Yasmin? So they write stuff here. I read it, and then I answer the question. Because I'm a teacher, right? I work at a school.
You're not a teacher.
I'm not a teacher?
No.
What am I?
A Bobby.
A daddy?
Daddy.
Yeah. I'm a daddy and a teacher.
No.
Mm-hmm.
I'm not a teacher. You're not a teacher.
Oh, man. What else, guys? Keep them coming. Yasmin, stop. Baba. No. All right, so just to give you guys some helpful hints in your studies, the test is pretty balanced. There's about equal amounts of chapter 8 as there is chapter 9. The chapter 8 material, if you guys remember, goes back to the LDRs, the LIRs, the Calvin cycle, reactants, products of each stage, the chemical equation for photosynthesis, um, factors that affect photosynthetic rates. Within that, you also have like the CAM pathway, which is how desert plants photosynthesize during different parts of the day. So the LIRs at night and the LDRs during the day. So that type of information, all of that is on there. After looking at the forms today, I could clearly identify that pretty much everything we covered in the slide presentation is on there. This means stop being difficult. All right, Stephen asks a question. Do we have to know the chemical equations for energy yield? Yes, in terms of you have to know how much ATP is produced at each step. If you're talking about cell respiration. So you want to know how much ATP is produced during glycolysis, how much it costs to invest. Yasmin, I can't.
I want to see him. I want to see your friends' pictures.
They're here. You see them? Okay. All right. So you got to know the amount. You got to know the amount of ATP at each step. So glycolysis produces 4, net cost of 2. Krebs cycle produces 2, 1 per pyruvate. And then electron transport chain produces 34 ATP because of the 10 NADHs. And those are each worth 3 ATP. And then the 2 FADH2s, which are each worth 2. So that would be 30, and 4 would make 34 during the ETC. And Yasmin is bored already. All right, so that's what you need to know.
No.
Caroline says, can you go over factors of the photosynthetic rates? All right, so like technically there's 4, but let's just assume that water is plentiful and available. So the 3 major factors that affect photosynthetic rates, you've got sunlight intensity. So obviously the brighter and more sunlight there is, more photosynthesis. That's good.
Yeah.
Yep, not a lot of sunlight. That's bad. Then you have CO2 concentration. So if the stomata are open, they can release O2 and take in CO2. Lots of CO2, good photosynthetic rates. No CO2, low photosynthetic rates. Right?
Mm-hmm.
Right. Mm-hmm.
Right.
And the last one is going to be temperature. So if temps are really, really hot or really, really cold, that's bad.
Yeah.
But if temps are just right, above 5 degrees Celsius, up to about 30 or 35 degrees Celsius. Very nice, very nice. Yeah. All right. Yeah. All right.
Don't open the door.
Gus says, hold on, Yasmin, we're answering questions. Gus says, why does FADH2 produce less ATP than NADPH? The reason, Gus, is that FADH2 doesn't carry as many electrons as NADH. So because it carries less electrons, it has less potential energy. So essentially NADH2 is NADH is a more capable electron carrier. Good night, Yasmin. Yeah, so that's that. Dory asks, what are the reactants of fermentation? So it starts with pyruvic acid. That's coming from glycolysis. So that's your reactant. And also NADH, the electron carrier, is a reactant. So those are your 2. That's it for that. No cost of ATP, but it also does not generate ATP. So let's see here. Sabrina says, where does the hydrogen ion concentration gradient occur? Depends if you're asking photosynthesis or cell respiration. So Sabrina, if you could clarify. Now, Sabrina, if you're asking about photosynthesis, and cell respiration. Okay. In cell respiration, the concentration gradient is occurring in the electron transport chain. So we're talking inner mitochondrial membrane into the inner mitochondrial space. So if you actually look in the textbook, there is an image. I'm not really sure what the page number is. I don't have a textbook with me. But you'll see where the H plus ions are abundant. It's not in the matrix. It's actually in the intermembrane space. So between the two membranes, between the inner membrane and the outer membrane, towards the outside of the mitochondria. Owen Cooper says, can you go over pyruvic acids in the Krebs cycle? So it's pretty self-explanatory, Owen. If you look at the Krebs cycle, you'll see that pyruvic acid is able to actually diffuse across the outer and inner membrane of the mitochondria, and that pyruvic acid can actually enter the mitochondrial matrix where it can be processed in the Krebs cycle. So the first thing that happens to pyruvate is it becomes oxidized. When it becomes oxidized, essentially, it's going to undergo a chemical reaction, a redox reaction with oxygen, and that is going to produce your first molecule of CO2. With the help of coenzyme A, it forms acetyl-CoA, and then it can start that citric acid cycle that goes through and produces more CO2 and ATP and FADH2s and NADH. So yeah, it goes through all those different conversions, and it's really all about the electron carriers in the Krebs cycle. So yeah, it does do some transformations, 2 carbon to 4 carbon and all these other things, but you guys don't really have to know those individualized steps. Just make sure you know that pyruvate is going into the Krebs cycle to help generate a lot of those electron carriers and help the formation of CO2 and the oxidation of said pyruvate. Dory, NADH is actually a reactant in fermentation. NAD plus is what's left over. It's more of a product. So remember, NADH is an electron carrier. So during fermentation, the electrons are acquired to form the lactic acid, to form the ethyl alcohol. So NADH is required. NAD plus is kind of the remainder. So that's the product, the NAD plus. Jack, so actually I just explained it. Owen was asking the same question. So pyruvic acid diffuses across the mitochondria. So it enters the mitochondrial matrix. It becomes oxidized, forming acetyl-CoA, and with the help of coenzyme A, which then it becomes another intermediate, becomes citric acid, and then it goes through a set of steps, further becoming more, undergoing more oxidation, a couple other conversions. All you guys have to know is what's produced during the Krebs cycle from the pyruvate. So that means the CO2, the NADHs, the FADH2s, and the ATPs. And make sure you guys know the numbers on those, the number of molecules. So 1 ATP per pyruvate, 3 CO2 per pyruvate, 4 NADH per pyruvate, 1 FADH2 per pyruvate. Double each of those for the 2 pyruvates from 1 glucose molecule. Good questions. Glad you guys are asking them. Very good. All right, definitely, Jack, go over the diagrams in the book on the Krebs cycle, just so you can be able to count all of the different products from it. So make sure you know the products, make sure you know the numbers of each, and you should be good to go. Make sure you know the location of where the Krebs cycle takes place. Angelica says, do we need to know all the reactants for the Krebs cycle, such as coenzyme A? I don't think so. I'm trying to remember. I mean, every form is a little different, but I'm almost positive that I don't ask any questions about coenzyme A. I don't think there's anything on acetyl-CoA. I don't think there's anything on citric acid. Pretty much, when it comes to the Krebs cycle, I just need you guys to know where it takes place, what the products are, and that it's aerobic. That's pretty much everything you need to know for the Krebs cycle. And the same thing for the ETC. You're going to want to know, how much ATP is generated, where it takes place, the final electron acceptor as O2, forming H2O. Like the really important stuff that I made sure that I highlighted in your notes in the slide presentation, as well as in the review guides that I gave you guys. Yeah, so Angelica, I wouldn't consider acetyl-CoA or coenzyme A as reactants of the Krebs cycle. Reactants of the Krebs cycle, you're going to want to know pyruvic acid. You're going to want to know oxygen is also an important reactant, because without the oxygen, there's nothing to oxidize the pyruvate. It won't form the CO2. And if it doesn't form the CO2, we can't get an oxygen and CO2 concentration gradient. So, yeah, that's a pretty big deal. Good questions. Keep them coming, guys. I see a lot of questions on cell respiration. That's really been the focus. Don't forget that there is information. About half the test is photosynthesis. So, hopefully you guys feel pretty confident in that. Make sure you're good with that diagram that we looked at with the chloroplast and all the individualized reactants and products. Go back over that. Go over the diagrams of your text. Make sure you know the products and the reactants of each stage, LDRs, LIRs, Calvin cycle, factors that affect photosynthetic rates. All that stuff is important. I feel that maybe you guys are asking more questions about cell respiration because maybe it's a little bit harder. If that's the case, no problem. Just don't forget about photosynthesis. That is on there. And in case you guys haven't seen it, on this YouTube channel, you guys will see that there are slide presentation video lectures for both cell respiration and photosynthesis. So go back and watch those tonight in your preparation for the exam. Go over the slide presentation. Go over all of your homework assignments, all your anticipation guides, concept checks. Go over all that stuff, all the review guides, everything. Look over everything, and of course, ask questions here within this video, or just do it as a comment on the classroom. I'll get to you guys eventually. All your questions, I'll try to answer as much as I can. I know this is pretty tough stuff. So, John, get out of this live chat. What are you doing here? Could somebody please explain why Koss is in here? I have no idea. They'll let anybody in these live chats, I swear. Security. Sabrina says the stomata opens up to take in sunlight and water, right? No, not at all. Sunlight is absorbed by chlorophyll. All right, big man, take it easy, man. Stomata opens up strictly for gas exchange. So what guard cells do is guard cells will use osmotic pressure with their large central vacuole. Remember, it's full of water. And what it could do is the guard cells can actually force open or force close stomata using that osmotic pressure from the large central vacuole. Remember, sunlight is specifically for the LDRs. Chlorophyll, which is located in photosystems in the thylakoid membrane, are going to be utilized for absorbing sunlight. So keep those things separate, all right? LDRs in the thylakoids, and then we have the LIRs, the Calvin cycle, and the stroma. And of course, guard cells are strictly for gas exchange. Chlorophyll, which is in the photosystems 1 and 2, strictly for light absorption. And don't forget what the sunlight is for. Sunlight specifically is for exciting electrons and photolysis, where the water molecule can be split. The H plus ions are retained. They're used for recharging NADP plus to NADPH, where it can be used as an electron carrier. That's a really important concept. So it's not that plants need the water, it's that they need the hydrogen plus ions from the water. So the O2 in water becomes waste, right? All that oxygen gets pushed out of the stomata as they're opened, and as they take in CO2, they release the O2. Any other questions? Keep them coming. All right, so far so good. You guys have had some good questions. Just so you know, depending on your form, I'm seeing about 50 questions or 51 questions on the test. Like I said, it's pretty balanced. There's probably I don't know, about 40 to 43 traditional multiple choice. And then there's about 6 or 7 of identifications and labeling. And then you've got one written response, which is 100% on your 2 labs from this unit. That would be the photosynthesis lab with the spinach leaves, the stomata and guard cell labs, and the cell respiration lab obviously was the balloon with the yeast and the sugar, the fermentation lab. So that's a big one. That question is going to be worth about 9 points, about 9 or 10 points. So very, very heavy. All right, y'all. What else you got? Yes, Angelica. Photosystems 1 and 2 are for absorbing sunlight. Do keep in mind of what that sunlight is for. So it's for the process of photolysis, where sunlight is actually going to be used. Well, it does a couple things. Number one, it excites electrons for the ETC. And the second thing, it's going to be splitting the water molecule. Remember, for the LDRs, the H plus ions are required as part of the electron transport chain. NADP plus can be regenerated into NADPH. So that residual oxygen gas that's left over just forms a concentration gradient. It's going to diffuse out of the chloroplast so that CO2 can come in and then O2 can go out. So yeah, photosystems 1 and 2. Now keep in mind, photosystem 2 comes before photosystem 1. I know it's a little strange and I don't want to get into the whole story, but they discovered them out of sequence and then thus they named them incorrectly, but it was too late to change it. So there's a diagram in the textbook. You guys can see it. I believe it's, I think it's 8.2 or 8.3. I'm not sure, but it's in there. Definitely go over that diagram. Steven says, will we have to label the parts on diagrams? Yes. I can tell you right now that of the 3, 4, 5 forms that are going to be circulating around tomorrow in the room, depending on your form, you'll have at least 2 diagrams, if not 3. One of the diagrams is a chloroplast. Another diagram is the mitochondria. Another diagram is the photosynthetic process as a diagram with the chloroplast. And another diagram is the mitochondria, the cytoplasm view of cellular respiration. So make sure that you go over all those, all the different handouts that we've gone over, all the different diagrams that we've done, all the supplementary stuff we did. It's all on there. Thanks for the question, Stephen. Good question. Dory says, wait, does oxygen go in and CO2 out? Actually, the other way, Dory. In photosynthesis, O2 is going out, CO2 is coming in. And of course, in the mitochondria and cell respiration, it's the other way around. So the O2 is coming in, the CO2 is going out. So really, the gases that are part of the biochemical pathways, they're opposite and inverse for cell respiration and photosynthesis. So like cell respiration, CO2 is a product and then O2 is a reactant. Well, in photosynthesis, it's the opposite. All right, CO2 is a reactant, O2 is a product. So try to keep track. If you remember the photosynthesis equation, then pretty much all you got to do is just kind of like turn the arrow around and you have yourself cell respiration. That goes for the gases, that goes for the products. It's pretty much inverse. Hopefully you guys know what the word inverse means, right? Opposite, and yeah, products become reactants, reactants become products. All right, good questions. I'm going to type something in the live chat here. Questions. And if you're watching this on replay, if you have questions on anything we're covering or something different, just go ahead and ask them, and I'll try to answer them at some point tonight or early tomorrow before I get head out to school. So I see a lot of the morning class are in the live chat. I don't see anybody from the afternoon class. Interesting. All right, it's nice to see we got a nice group of kids in here. We've got about 7. I think I've seen it peak at about 9. Excellent. Any other questions, y'alls? Trying to think, was there anything on the test I forgot to tell you guys about? I don't know. I gave you guys the format, told you how many questions there are, what they're going to be like. It's not easy. I think cell respiration is challenging. It's a very difficult concept to grasp. Photosynthesis was obviously a little bit challenging as well. Hopefully, you guys are pretty comfortable with them, though, for the most part. Gus asks, where are the enzymes for the ETC found in prokaryotes? In the cell membrane. In the prokaryote cell membrane. Now, the reason that is, Gus, is prokaryotic cells don't have mitochondria. So because they don't have plastids, because they don't have organelles, specialized internal structures within the cell, they don't have the ability, to do any type of traditional transport chain, no Krebs cycle. They don't have that because they don't have mitochondria. So that being the case, they just kind of do what they can within the membrane.
Do you want him to work on the tank tonight or wait until morning?
Tonight.
Tonight?
Yeah.
Yep.
I mean, all right, y'all, I'm back. Sorry about that. Had to tend to something. All right, we're about 20 minutes in. I was hoping to be on here for at least 30 minutes. So that puts me at the 30-minute mark almost. If anybody has any other questions, just go ahead and ask. Type them out in the live chat. Or, again, if this is on replay, just ask in the comment section. I'll try to get to you guys as soon as I can. And also, if anybody has any questions at any point tomorrow, you can come in and see me. I usually get to school around 7:15. So if you want to ask anything, just come in a little bit earlier in the day, or if you're in the afternoon class and you want to ask questions, just come in right after lunch, at the end of your lunch period, if you want to ask or whatever. Yeah, just pop in at any time tomorrow before the test. All right. Any other questions? We're at the 29-minute mark. Don't see anything going on in the live chat. Maybe everybody's gotten a chance to ask all their questions. Maybe you guys are just too smart and you already know everything. Hopefully that's the case. Oh, and if you are watching this on replay, the live chat is available. You just got to click on the live chat button. It'll show you everything in real time, all the questions that were asked. But what I like to do is I like to reread the questions verbally and through audio so that you guys can hear them and then get the answers to them. All right, things are pretty calm and quiet in the live chat. I think we'll just leave it at that. If you have any more questions, and this is on replay, then just go ahead and ask in the comment section below. I'll try to get to them, or post the question to the most recent announcement on Classroom, and me or maybe another student or somebody can offer you an answer between now and the end of the night, or I'll get to it tomorrow morning. So, all right, that's it. That's about 30 minutes. That's what I could do for you guys. Hopefully, you guys kind of have a better understanding and you clarified some of the misunderstandings and some things that were confusing. Hopefully, it was helpful. I hope you guys appreciate it. I'd love to see you guys do well on tomorrow's exam. And hopefully, that's the case. So, work hard, study hard, reread parts of the text, go over all of the assignments from the last 2 units, go over all the labs and materials, go over all the relevant vocabulary, all the review guides. Just make sure you're familiar with everything and you should be fine. Your hard work will pay off, I promise. And hopefully you guys do well. And I'm cheering you on here. I want you guys to do well and hopefully you have a good score and it's to your satisfaction. All right, that's it for this one, guys. Thank you for tuning in and asking your questions. I'll see you guys tomorrow.